Thanks for having me, Matt. I'm really happy to be here, and good afternoon, everyone, or good morning, depending on what time zone you're in. This morning, I'm going to be talking about some of our research that we've been doing to optimize yields of corn planted after a cereal rye cover crop. And, oh, that's not good. Okay, there we go. So I wanted to take you back a couple of years to a webinar that I gave in February, two years ago now, and just bring up what we've been researching. So we know that winter rye is a host of pathogens that cause seedling disease on corn. And what happens is that when we terminate the winter rye, spray it with a herbicide such as glyphosate, as those roots die, they release these Pythium, predominantly Pythium, but also Fusarium, but we focus on Pythium, into the soil. And what you'll see is this increase in the population, and then slowly that population will decline and go back to background levels. So what happens is if we plant corn too soon after we've sprayed the rye with the herbicide, we're planting corn into a period of time when the populations of the Pythium are very high. So it's almost like planting your corn into a petri dish full of Pythium, right? And so that increases your risk for seedling disease. And so here on the right, you can see a picture of a corn seedling. You can see that the mesocotyl is rotten and that the seminal roots and the radical root also have some seedling rot on that. So this means that winter rye acts as what we call a green bridge for seedling disease. As those pathogens are released from the roots of the rye and they increase, they're looking for a host. And if we planted corn, when those Pythiums are hanging around looking for another host, then they will infect the corn. And so what we want to do is we want to try and plant the corn when those Pythium populations have decreased back to background levels, and therefore we're at lower risk for seedling disease. So in our work that was funded by the Iowa Nutrient Research Center, we were able to prove that this was indeed the case. So here you can see where we always have a no-cover crop check in our experiments. And you can see that when we had that in the no rye check, and then the rye where we sprayed either 25 days before planting or 17 days before planting, that there was significantly less disease than when we planted that corn within a week of terminating that rye. So our recommendation has been to wait at least 10 days to plant corn after terminating a winter rye cover crop. Okay, before I move on a little bit, I just want to talk about why seedling disease. Why are we so fascinated with seedling disease on corn in my lab? And so this picture just shows you what seedling disease can do. It can kill the seedling, but more importantly, what can happen is that if we get any damage to the mesocotyl or the seminal roots or the radical, that can affect the early growth of that seedling. And so here in this picture, you see a stunted seedling, and I'm sure all of you know that seedling often becomes a weed, and it either produces a very small ear or no ear at all. And if it's not producing an ear, it's not contributing to yield. So on the left, we have a diagram of a corn seedling. So just showing you that the mesocotyl, that pipeline that's taking the nutrients from the seeds to the developing crown of that plant. You can see the nodal roots emerging. This is a V1 seedling. And those nodal roots will kick in around about any time from kind of V3, V4 onwards. By V6, the plant is completely dependent on those nodal roots. But up until then, it's very important that we keep that seedling root system healthy. But we have a problem, right? Because almost every year we get cold, wet springs. Okay. And the problem with cold, wet springs is that they prevent access to the field to terminate the rye. Okay. And then also when it's cold, the glyphosate's not effective. And so we can go out and spray, but we might not kill the rye. Okay. And so this leaves farmers with two options. One of them is that they can wait until things are warm and dried up and then spray the rye and then plant the corn, 10 days plus after terminating that rye. But how many farmers like to just sit and wait for things to warm up, right? After winter, we're always antsy to get into the field and get things planted and get things on the way. And so a lot of farmers have been planting their corn and then spraying rye a few days later after planting corn. And so this practice is referred to as planting green. Okay, but based on our research, we have a lot of concerns about planting green. And because we think that could increase the risk of seedling disease. So what I'm going to talk to you about is some research that we've been doing looking at planting green. And this is a photo of one of our field trials. And this project, we're working in collaboration with Practical Farmers of Iowa on this project. And the project is being funded by USDA NIFA. So in this project, we have a lot of extension that will be starting this year, but we also have some research activities. And so the research activities we have is an experimental plot field trial. So those are small plots, 5 rows. You can see a picture there. We also have a study where we have microplots. I'll be talking about the experimental plot and the microplot data that we collected in 2019 and just sharing that with you and sharing some ideas about how, thanks to Practical Farmers of Iowa, are working with 4 farmers and doing on-farm trials. So in our experimental plot field trial, we have a number of different treatments. So first of all, it's a planting green treatment, right? So I'm going to focus on when are we spraying or terminating the rye. So we're either terminating the rye or spraying the rye 18 days before planting, 3 days before planting, 6 days after planting, 12 days after planting, and then of course we have a no-cover crop control. Now superimposed on top of these, we are also looking at nitrogen. So most of those treatments have received 50 pounds of UAN at planting. That was 5 pounds in the furrow, 45 pounds in a 2x2. And then we come back and side dress with 130 pounds of nitrogen at V4. But we also have another treatment where we're looking at applying 120 pounds of UAN pre-plant. So that was applied when we terminated the rye, 18 days before planting, and then we come in and side dress it at V4. This experiment is done on a corn-soybean rotation. It's no-tilled, and the rye was drilled in September. So you can, this is a picture from this year's trial, and you can see that we had really good growth of the rye. So if we think back to what our hypothesis is, you can see the brown curve here. That's showing you that when the rye is terminated 18 days before planting, this is what we expect the Pythium populations to be doing. Okay, so increase and then decrease back to background levels. And then obviously, 3 days after planting, 6 days after planting, 12 days after planting, we shift that curve to the right, okay? The corn all gets planted on the same day. And so we're looking at how these different Pythium populations might affect seedling disease and growth of the corn. So this is our data from 2019 and 2020, our biomass, and it's probably not surprising to anyone out there that the later that we terminate the rye, the more biomass we have. And so, that's pretty clear there. And you can see that 2020 has been a very good year for rye cover crops, and we had some excellent growth there. We also take stand counts, and this data here is the stand count from 2019. And apart from this 18 days before planting, where something a little bit odd went on there, in general, we see good stand counts, except in this 12 days after planting, where the stand counts may be slightly reduced. Obviously, 2020 we'll be able to see if we see that same data. But probably more importantly or more interesting is that when we look at the plant height in these treatments, although everything's at the same growth stage, the plants where the rye has been terminated 12 days after planting are taller compared to the plants in the other treatments, and particularly in the no rye check. And that's not really surprising, right? These corn plants are being shaded out by that rye, and all the biomass there, and so they're really stretching up to get to the light, and so they tend to be taller. And they also tend to be a lot skinnier than the plants in the other treatment. Of course, we're interested in the seedling disease, and so just as we expected, we see more seedling disease when that rye is terminated too close to the corn planting or after the corn is planted. So in the 12 days after planting, we saw the most disease, more disease when we have the 3 days before planting, and then that planting green, we saw significantly more disease than where we either had no cover crop or we terminated that cover crop 18 days before planting. So that's our concern, and we were able to show that. Of course, before we get to yield, so we always look at the population of Pythium that we have in those corn seedlings. And so as you can see, with the greater yield, we also detected more Pythium species, firstly in the radicle, 6 days after planting, and then in the seminal roots in the 12-day after planting treatment. And that probably has something to do with the way that corn seedling is developing, right? So in the 6 days after planting, the radicle was probably at a susceptible stage to get infected. The seminal roots were still emerging, so we don't see as much Pythium on those. And then when we get to the 12 days after planting, that radicle's pretty mature and growing, so we detect less Pythium, but we find more Pythium in the seminal roots, which are now at that susceptible stage. Yield, as we saw, we just got reduced yields in our plot where we planted the corn following rye, except in the one plot where we'd applied the nitrogen at pre-plant 18 days before planting the corn. So when we sprayed the glyphosate, and that was probably a little surprising to us. So we'll be interested to see this year how those results look. As I mentioned, we're doing on-farm trials, so I don't have any data to share with you from this year. But this is one of our collaborators, Kevin Hull. We were at his farm in June, doing stand counts and collecting seedlings, and so you see Kevin here helping. And then Jyotsna Acharya is an assistant scientist in my lab, and she really leads these studies and collects the work and summarizes all the data and gets the publications written. So the other thing that we've been doing is microplot studies. And the reason why we've been doing the microplot studies is because we were wondering how nitrogen affected disease risk. And so Jyotsna came up with this really neat idea about putting out some microplots. And so this is what our microplots look like. This is a study that we have, and you can see that we have no rye control, okay? And then you can see the rye residue on the left, okay? And then you can see those flags. We have white, yellow, pink, and red flags. And those flags are demarcating the plots. And basically that plot is 15 to 20 plants long. And then we go out there and we either apply UAN, so 10 pounds of nitrogen per acre, Miracle Grow, which of course contains nitrogen, but also P and K and some micros. And then we have a water alone control and a no treatment control. And we've been seeing, so what we were thinking was that maybe with a lot of biomass, the nitrogen's being immobilized, and so it's not available to that seedling, and that seedling's really struggling to get out of the rye, and so it becomes more susceptible. And so we were expecting to see less seedling disease where we were applying nitrogen to those plants. So if we look at some of the, this is the data from 2019, but what we saw was that in the corn that was planted after no cover crop was taller, it was bigger and heavier in terms of shoot dry weight, and it was also greener. And then, but it had less root rot compared to the corn that was in the rye. So just as we would expect. Now, when we go in and we look at how those treatments affected the growth of the corn, so this is a picture of the plots from this year, and I think that you'll agree that there's some striking differences there. You can see that the plants are greener and taller in some of those microplots. And then the data that I'm showing is from 2019. But what you see is that, first of all, you know, in the plots that receive water or UAN or Miracle Grow, the plants were taller. The plants were also heavier or bigger where they received those treatments. And where we received that extra nitrogen from the UAN or the Miracle Grow, the plants were also heavier and more sturdy. If we split apart that into the rye and the no rye treatment, you can see that in the rye treatment, we had a lot more effect of those treatments on shoot dry weight than we did in the no rye treatment. And so that rye, I mean, as we all know, is definitely affecting that early growth of the corn. We also took readings to tell how green the plants were. And so we do those meetings with a SPAD meter. That's a picture there showing you just how we hold a piece of the leaf and then just measure the greenness. And in 2019, we collected those readings at V5 to V6, at V8, and then around about V10 to V12. And so on the left here, you have a figure that shows you with the no cover crop and the rye, and it shows you the cumulative SPAD readings. So that was kind of the three readings over time. For those of you who are familiar with cumulative aphid days, basically the same way of calculating cumulative SPAD readings. But what you can see here is that particularly in the rye treatment, that the plants are greener where they received the water or the UAN or the Miracle Grow. What was very unexpected in this trial was that we had more root rot where we applied the nitrogen, and particularly the Miracle Grow. And you'll remember at the beginning that our hypothesis was that these seedlings are struggling because they're not getting enough nitrogen, and therefore they're more susceptible to disease. And so this was a big surprise to us that we have more seedling disease, with our UAN or our Miracle Grow. Once again, on the figure on the right shows you that if we split that apart into the no rye versus the rye, that really we have a bigger difference between the treatments in the rye treatments compared to the no rye treatments. So that was very unexpected, and we've gone ahead and repeated this in 2020. There's been a few differences. 2019 was very wet, and so we didn't get in to apply our treatments until around about V3, and then we sampled these seedlings at V4, whereas this year we were able to get in at around about V1 and do 3 applications of treatment, so approximately every 5 days, and then sample our seedlings. And so I'm very anxious to see what data Jyotsna has collected this year. So we're still waiting on that. Seedlings have been sampled, and now we just need to capture all that data. Obviously, in these microplots, we can't do yield. So we look at yield components, and very clearly, you can see that those plots that didn't receive any treatment, that their ears, the length of their ears was shorter, and then also that their kernel weight was significantly lower. And so that would indicate that probably lower yield, right? This is just some pictures from 2020. I just wanted to show you a close-up of those microplots. So, the top left, you can see where we don't have any treatment. The top right, we have the water. You can see that those plants are greener compared to the plants in the side rows. And we suspect there that the water, if you remember that we were applying water to these plants every 5 days. And so that probably helped with that 2x2 nitrogen that was nearby, just to get that to move through the soil profile and for these seedlings to pick it up. With the UAN, the plants are greener. And then with the Miracle Grow, I think you can see that the plants are greener still from that. And this was on the rye. And then if you look on the soybeans, a little bit harder to see a difference between those treatments. Miracle Grow, still greener, but the other treatments a little bit more difficult. So the summary of our data from 2019 is that planting green in our small plot trials affected corn growth and development. We saw lower stand counts. The plants were taller. We saw more root rot, and we were able to detect higher populations of Pythiums in that root. And so that kind of goes along with what we're expecting. We're very interested, obviously, to see what happens in the on-farm scenarios, because it's not unusual for on-farm scenarios to be different to what we see in the small plots. And then, spoon feeding in our microplots definitely improved the vigor of the corn planted after rye. However, we saw more root rot on the corn at V5, which was a little bit of a surprise. And we have to figure out how to explain that. Before I end off, I just want to mention that this study is going national. So I am part of a national grant that's being led by Stephen Mirsky in Maryland and Chris Rehborg-Horton in North Carolina. And it's a big grant on precision sustainable agriculture. And I'm involved in Common Experiment 2. And what we're going to be doing is we're going to be looking at the effect of cover crop termination timing on slugs, on insects, diseases, weeds, and then also the performance of corn. And so this study is being funded by USDA NIFA, and these are the states that are taking part in the study. You can see me up there. I'm leading the disease part of the study. John Tooker from Pennsylvania is leading the insect side of the study, and Colleen Chase from Florida is leading the weed side of the study. And I'm really excited about this study. All of our work that we've done looking at seedling disease on corn following a rye cover crop has only been done in Iowa. And so I'm very excited to see what happens elsewhere in the US under very different conditions. So I'd like to say thank you very much to Iowa Learning Farms for having me today. A special thank you to all of you who joined for the presentation. And I'd be happy to take questions. And while Matt is getting those ready and you are asking questions, Hillary asked me to remind you that if you would like a continuing education unit from listening to this webinar, that please can you send the following information to her. There's her email address there, hepierce@iastates.edu. Remember your full name, the name you entered to watch the webinar, and then your ID number, so that she can get that to you. And then lastly, I just want to mention that next week is the first webinar in a series of webinars for cover crop boot camp. So Iowa Learning Farms hopes that you'll join them on that. So thank you for your attention, and I'm happy to take questions now, Matt.
Okay, great. Thanks, Alison. We've had some come in. So the first one, have there been any studies using kind of polyculture cover crop or cover crop mixtures, or have you really just looked at monoculture of winter cereal rye?
That's a great question. So we predominantly focus on cereal rye just because it's the dominant cover crop right throughout the US. But we did do a study a couple of years ago funded by Iowa Nutrient Research Center where we compared rye and camelina on corn and soybean. And so, but once again, that was just a monoculture of camelina or a monoculture of rye. So no, I have not done any research looking at that, and I am not aware of anyone else who has looked at mixtures, particularly from the disease point of view, right?
Great. Maybe you can go back to the previous slide. Somebody wanted to reshare the CCA information. Oh. Okay, next question. Was the corn treated with a seed treatment in the study on Pythium infection?
Yes, so all our corn is treated with a seed treatment. We still see Pythium. If we didn't use a seed treatment, I'm afraid that we wouldn't get any corn coming up. So, yep, seed treatment on it.
Okay. Is it fair to assume that Pythium would affect soybean in a similar way, not just corn?
That's an awesome question, because we know that the Pythium that infects corn also infects soybean. And so actually the study that I just mentioned where we use camelina, we had soybean in the rotation there, and for some unknown reason, soybean doesn't get affected by Pythium after the cereal crop of rye. So soybean is an awesome crop to plant after cereal rye. It doesn't seem to care. It just gets on and grows well and yields well. So, okay. Yeah.
All right. Does increased Pythium necessarily lead to reduced yields?
No, not necessarily. So, I went in that first study that I spoke about, where I did the webinar a couple of years ago. In our first year, even though we saw more disease, we didn't see a significant reduction in yield. It was only in the second year of that study where we got a significant reduction in yield with the increase in Pythium. So no, it does not happen every year, and I'm sure it does not.
Oh, shoot.
Yeah, we think it might have something to do with biomass, because in that study we had a big difference. So yeah, we're just, no. Just because you have Pythium there does not mean you're going to get a yield reduction.
Okay. The next one, could the increased root disease in the higher amount of spoon-fed nutrient be related to less association between the corn root and soil biology?
That's a good question.
Kind of, I guess there was a little comment. Some of these associations are beneficial in aiding the plant to defend against pathogens.
Yes. Yeah. That's a good idea. I guess I know Jyotsna and I and other people on this project, Keith Kohler, Tom Casper, Stefan, Galen's from Iowa Learning Farm. I mean, that's a good idea. I haven't thought about that, and we're still trying to figure out why we would get more disease. And I'll have to. I can't really answer anything. It's a good idea, and it's something that we should bring up and think about. Maybe we could look into that. Okay. Good idea. Thank you.
Next one. Can you make any projections on these practices for organic growers? With untreated seed, how many days before planting would you propose terminating the rye cover crop? Well, and actually I might add in, because the next question also might relate. Would you expect to see similar results if the rye was terminated by alternative methods, i.e. a roller or a mode? So that I don't know, I bring that up just because it might play in for the organic growers as well.
Yeah. So I think I don't know a lot about organic corn growing, so hopefully I don't mess this up too much. But I think one of the things with Pythium is it really likes cold, wet conditions, and so I suspect that the organic growers are maybe planting their corn a little later than commercial growers when the soils are warmer. And it's possible that when the soils are warmer and drier, you wouldn't run into this problem with Pythium. And so you could maybe get away with without using a seed treatment in that case. I'm not sure that I answered your question correctly, but and then as regards the rolling or the crimping, yeah, I don't know. I mean, as those roots die and those tissues decompose, they're going to release the Pythium. And I guess I don't have a good understanding about how fast the roots die and decompose with glyphosate versus a roller crimping, right? With roller crimping, if they take longer, then what you might imagine is that there's still an increase in Pythium, but it's probably a slower increase. That curve is flatter. I know how everyone likes to hear about flattening the curve at the moment, but that may flatten the curve. If the roots die quicker, then we might get a very sharp spike, right, in that curve where those Pythium populations are up and then they're down again. And so that means there's a very short window where you probably don't want to plant. So I guess I can't really answer either question very well. Those are just some thoughts. Yeah.
Great.
Sorry.
Okay. Were corn planting dates between the cover crop treatments generally the same?
Yes. So all the corn was planted on exactly the same day. So that took a little bit of agronomic gymnastics for Keith, who was, we had to kind of figure out when we were going to terminate our rye, when was our goal planting date, and then we would terminate our rye around about that date. So we tended, our corn got planted around about the first end of April, first week of May. And then we would figure out our termination dates from that, and then just hope that it wasn't wet and we could get in the field and then apply the glyphosate.
Okay. The next one, do multiple years of rye cover crop change any of these dynamics? For instance, improving the biology of the soil?
That's another great question. So I guess the fields on that we're working on have been in continuous rye corn soybean rotation with rye for several years. But, and I am not sure if Keith is on the webinar, but if he is, maybe he can type in. We definitely always plant the rye after soybean, but I'm not sure that we plant rye after corn every year. So that would mean that there would be a skip year, right? So we wouldn't have rye every single year. And you're absolutely right that that's going to affect our soil biology. And so it could easily, it could have some effect on what we're seeing. Another reason to go to on farms, right? Where
Was there a statement about using 180 gallons of nitrogen? Or is it 180 pounds?
100, I think the total nitrogen we apply to our corn is 180 pounds.
Okay.
And yeah, so most of the time we're putting on 50 pounds at planting, and then the balance 130 pounds at V4. Except for that one treatment, right, where we put 120 pounds on before planting, and then 60 pounds at V4. But okay. Yeah, so we put a total of 180 pounds on. Okay,
so maybe just a little bit above the MRTN, the ISU corn nitrogen rate calculator rate.
Yes.
Yep.
Yes.
Okay. Any assessment of possible allelopathy from rye affecting corn development?
Another great question. And that's something that my lab is starting to get into. Yes, I mean, when we first did this work, we were interested in what was causing the yield drag. And so we came up with the Pythium. But then from speaking to folks like this, right, that brought up a lot of other questions. So one of them being allelopathy, and then the nitrogen and the biomass. And I mean, so seedling disease is not the only reason for the yield drag. There's other things going on. And last year we had funding from the Iowa Nutrient Research Center where we started to look into allelopathy. And we did kind of just a little test in the lab using Petri dishes. And we were able to show that under certain conditions of allelochemicals that we saw more severe seedling disease with the Pythium pathogen. And so, this is a system, right? This whole rye cover crop affecting corn is going to be a system. There's lots of different factors, and we're trying to tease about all those factors. But yes, I agree that allelopathy is probably playing a role in all of this as well.
All right. All right, that is the end of the questions. If somebody has a burning question, please type in quickly. Otherwise, definitely want to thank Dr. Robertson for her webinar today. And again, the CCA information is on there, and I would encourage you all to get on for the cover crop boot camp next week. Looks like we have a couple of great speakers to talk about that. So if nothing else, thank you so much for joining. I wish you all a great afternoon and a great holiday weekend. So thank you very much.
Thanks for having me, Matt. Thanks for joining us.